Hello everyone, welcome to our second short review on innovative cancer immunotherapy with MACE A3 mRNA cancer vaccines. So before getting into this review, we have to know what a cancer vaccine is. Cancer vaccines aim to stimulate the immune system to recognize and attack tumor antigens. So how they work is, first of all, we have to extract the RNA from the patient's tumor tissue. This RNA contains genetic information about proteins produced by tumor cells. After that, the extracted RNA is administered to the patient. The modality of this administration, we will come to it later in the presentation. Immune system activation occurs once this is done. As RNA is taken up by APCs and they process the RNA and present fragments of the tumor specific proteins on the surface. This causes T cell activation, which destroys abnormal cells presenting this. So, as the tumor antigens become activated and they proliferate, T cells recognize that and it destroys the tumor cells that express the same proteins. Hence, this immune response can lead to elimination or reduction of the tumor. This principle has been shown quite evidently in this study. So, let's get into it. Melanoma associated antigen 3, that is MACE3, is a common tumor associated antigen found in melanoma. Now, T cells recognize MAGE-A3, which enhances its potential as a target for cancer vaccines, as I've told already. And since MAGE-A3 is minimally expressed in normal tissues and only during specific developmental stages, it does not trigger autoimmune actions. Lipid nanoparticles based on DMKD can encapsulate mRNA molecules and serve as a modality of, like, they can preserve it and serve as a modality of delivering it. Now, this section contains the basic methodology as well as the results. Methodology is given separate in this article, but since we are briefing it, I'll directly get into the results. So MAGE-A3 mRNA using in vitro transcription method was synthesized and then the plasmid was constructed. The MAGE-A3 gene sequence was derived from CT26 cells, which is a mouse colon cancer cell line, and it was inserted into the plasmid. And then the successful insertion was confirmed. After that, DMKDPS lipid nanoparticles, they could deliver proteins to immune cells as per previous studies. So the same formula was employed to assess the anti-cancer immunotherapeutic efficacy. They were formulated inside DMKD and transfected into mouse macrophages. And then successful transfection was analyzed using RT-qPCR in Western blot. Now, as per several studies, multiple LNPs were seen, and then out of these, basically DMKD PS LNPs maintained their structural integrity and successfully delivered mRNA even after 16 weeks of refrigeration, as compared to others which showed a decline in mRNA delivery efficiency over a period of time. Now, to evaluate whether MAGE-A3 mRNA anti-cancer vaccine could suppress tumor development, these tumor cells from mouse colon cancer cell lines were implanted into mice that had been immunized with the mRNA cancer vaccines 2 weeks prior to it. Now, tumor size was monitored 4 weeks after that. And despite vaccination, CD26 tumors formed, but the growth rate was generally slower in vaccinated mice. This is a significant finding which we can find over here. By week 4, tumors in DMKD PS mRNA vaccinated mice were approximately 1.1 times larger than in week 3. But in untreated control mice, there was a threefold increase in tumor size, which is what is inhibited by the vaccine. So body weight measurements showed a slight decline, but this is not a significant toxicity. That is, it's not significant weight loss as such. Now, histopathological assessment was done to skipping that, as we'll directly go to survival rate. So, to evaluate the effect of MAGE-A3 mRNA anti-cancer vaccine on inhibiting tumor mets, CD26 were administered by the tail vein of mice. And they were already administered with mRNA formulated in various LNPs. Now, mice administered MAGE-A3 mRNA and DMK-PPS LNPs exhibited significant longer survival with a stronger p-value compared to untreated control group. So here you can see in the graph where DMKDPS stands as a superior delivery LNP as well as the efficacy of the mRNA vaccine is quite evident. MAGE A3 mRNA encapsulated in various LNP formulations was intramuscularly administered to tumor-bearing mice. And then doxorubicin was also given, which is an already established anti-cancer drug, to compare the therapeutic efficacy. Tumor growth observation was terminated on day 22 and tumor size in untreated control mice exceeded 10% of the body weight. That's why it was stood. Now, DMKDPS formulation demonstrated anti-tumor efficacy compared to that of doxorubicin. This is a very significant finding. But mice treated with doxorubicin experienced weight loss, while those treated with our mRNA vaccine gained weight during this study endpoint. This could be also due to like tumor growth as such, but then this is like lesser side effects for the vaccine treated mice. And also following vaccination, all cancer vaccine formulations led to an increase in immunoglobulins. DMKD-PS formulation enhanced the levels of IgG2A and IgG2B. That is critical for anti-tumor immunity. That is, so we can say that increase in immunoglobulins critical for anti-tumor immunity was found post-vaccination, which meant that the vaccine signaled a strong secondary immune response, which is beneficial to us. Thank you everyone for listening.